Good evening, everyone. It's a pleasure to see you here, especially in a day like today. We have more resilient people coming in. I'll do a slow introduction so that we have everyone in-house and they can just run in. So, my name is Rui Costa, and I'm an Associate Director and CEO at the Zuckerman Institute. And it is my pleasure to welcome you to this evening that is part of our Stavros Niarchos Foundation Brain Insight lecture series. So tonight's event is a continuation of this strong series of outstanding lectures, and today we're going to touch a topic that I think will make all of you be glad to be here. It's a very exciting topic pointing to the future and how neuroscience and psychology can help us in education, development, and a lot of times we talk about translation of science to our everyday lives, and we mostly focus on, for example, medications. But a lot of the benefit of research like the one you will hear about today, I think will impact or will translate to our everyday lives, our schools, how we organize society, how we organize our own lives growing up. So we're very thankful to the Stavros Niarchos Foundation for their continued support in this lecture series. They also sponsor a complementary teacher-scholar program so a lot of what we talk about at this lecture series goes then into materials that are lectured by teachers at schools. So this is the real translation. There's the impact of the research into the lives of people at schools. So I would like to thank you once again for your continued support on this and for being here in a very snowy day. And I would ask everyone if you please join me in thanking the Niarchos Foundation for their support. So this is an amazing time at Columbia University, as you will see in neuroscience. We have the Zuckerman Institute that is now fully open and with 47 faculty in-house, and as of today with more than 150 associated labs across campus. Some of the research that you'll hear about today happens at the Jerome L. Green building where the Zuckerman Institute is hosted. And the vision of this institute is that to tackle how our brains work, how our memories are formed, how we feel, how we decide, is not a simple job. So it's a tall order. And in order to do that, the university thought we need to combine all minds from different disciplines that are thinking about this problem. Some people from psychology, from engineering, from physics, from neuroscience, from neurology, from psychiatry, from radiology, need to be together to try to discover things, fundamental things about how the brain works and how we can ameliorate it in situations in which it goes wrong. So we continue to be honored by Mort Zuckerman's gift, by the Jerome L. Green Foundation gift, by the Niarchos Foundation support in all of these. And we continue to look for partnerships and for people that want to be with us in this journey of discovery of one of the real most important conquests of our time, the understanding of the brain. So in order to bring you into what is known now, we have the luck, really, of having Dr. Tottenham here tonight. And her research focused on the development of the brain, but especially on the development of the neural circuits that underlie affective behaviors across childhood and even adolescence, and with the emphasis on what are called limbic-cortical connections. And one of the major focuses of this laboratory is to establish what is the normative brain development. I mean, isn't it amazing that we know so little about how the brain develops and how the interaction with the environment under conditions of stress? Sometimes we are not going to hear so much about that tonight, but just during normal development, what's the normative situation? How can we know if this is going right or wrong? In many other fields, like for example in heart studies, it was only after a lot of evidence of how the heart normally behaves that now we can say, even with a little iPhone, sometimes people can diagnose, oh, there's something wrong, I should go to the doctor. Many times we don't know that about the brain. And so Neil Tottenham is a professor here of psychology and the director of the Developmental Affective Neuroscience Laboratory. She's really a world-class researcher, but also a wonderful speaker. If you have not had the opportunity, we're in for a treat. She has given many lectures nationally and internationally, and she has received many awards for her work. I'll just mention a few. She's a fellow of the Association for Psychological Science, and she has received the National Institute of Mental Health Biobehavioral Research Award. She has received also the Innovative New Scientists Award, that's called the Brains Award, the American Psychological Association's Distinguished Scientific Award for early contributions to psychology, and the Developmental Science Early Career Research Award. She's a wonderful lecturer, a wonderful scientist, but also an absolutely courageous person. She was the first here, and she was like, we're gonna do this. So please help me in welcoming Nina Tottenham to the podium to hear her lecture. Thank you.
Okay. Good evening, everyone, and thank you so much for coming out in this weather. A special thanks to the teachers and students that trekked all the way here. I'm really excited to tell you about some of the things that we've been learning about human brain development. So the work in our lab really addresses this very old question in psychology, which is how do we explain this enduring link between early experiences that we have and our behaviors later on in adulthood. And as you can quickly appreciate, in the human, that link lasts for a very long time, if not a lifetime. And so the approach that we take is to study brain development to understand that glue. So how the early experiences that we have might influence the course of brain development, and that would therefore tell us something about the behaviors that we can expect to see later on in maturity. So today, I'm going to be focusing on very much focusing on the development of the amygdala and its connections with the prefrontal cortex. So I think all of you are very aware, but here we have the front of the brain and the back of the brain. And deeply set in the brain are the amygdala nuclei, which is this evolutionarily old set of nuclei that live in the brain. And we often hear about the amygdala as being involved in our threat responses. And the amygdala definitely is responding to threat, but it responds to many things. And so the best way to describe what the amygdala does is it helps us direct our emotional attention to what's important. Threat is very important, it turns out, and also for emotional learning, so learning about the relative safety and danger of the environment. Now, in adulthood, the amygdala has strong two-way connections with the medial prefrontal cortex. So we can think about these connections as a superhighway between the amygdala and prefrontal cortex. And the prefrontal cortex collects information from many regions of the brain, including the amygdala, that by and large serve the purpose of helping to regulate or dampen the overarousal of the amygdala. And as I'm going to be describing, that superhighway between the amygdala and prefrontal cortex takes a very long time to develop, especially in the human. So we study this system during development, and by studying the unfolding or the construction of this system, we think that we can better understand mechanisms that result in individual differences in our behaviors. So if we look at individual differences in emotional behaviors, or another way to say that is if we look at mental illnesses that are associated with emotional difficulties, they tend to show this peak age of onset at the beginning of adolescence. So our approach to understanding these individual differences that emerge at the beginning of adolescence is not to look at the beginning of adolescence, but instead to look at the antecedent period, to understand what's happening before we get to that point in adolescence. So another way of saying that is we believe that there are sensitive periods of brain development that are happening when the environment is having a particularly large impact on how we're developing that gives rise to all of these individual differences that we see in adolescence. So what's a sensitive period? A sensitive period can best be thought of as these moments in development when neural circuits show heightened plasticity or heightened changeability by the environment. So we can think of, if we have age here along the x-axis and neural plasticity on the y-axis, there are these moments or windows of plasticity that seem to open up, so to speak, when the environment has a particularly large impact on the way that that system is going to function at points thereafter. So that once the system switches from a plastic to a more consolidated system, it's going to be harder for the environment to shape things at this point. But the behaviors and the brain outcomes that we see at points thereafter should be a direct reflection of the impact of the environment during these sensitive periods. So that's the main working model of a sensitive period. So if we look at the prefrontal cortex, many papers across. Then there's a little bit of change at points thereafter. Puberty gives us another bump in size, but all of those changes are really dwarfed by what happens in the first year after birth. So the point that I'm making here is that structurally the amygdala is showing a lot of development very early in life. Our research asks the question, what are the functional changes of the amygdala that rest on top of these structural changes? So we really want to look at reactivity of these systems. So to do that, we, for example, bring in a wide age range from 4 to 22, and we can scan individuals in an MRI scanner. So this is a safe way for us to look at typical brain development in healthy children. And we want stimuli that are going to be good at activating the amygdala. So we use fear faces a lot because it turns out that when we look at somebody else's face making a fearful face, that's a really good way to activate the amygdala. And because we're scanning participants that include really young children, we want to use a stimulus that's relatively innocuous, right? So we don't want to use shock with young children. So fear faces are pretty good at doing the job. So we present these fear faces to all of our participants from 4 to 22. And what we observe is that there is a lot of activity in the amygdala, but the amygdala shows decreasing activity as age increases. So I'm pulling out these data here to show you what I mean. Here's age on the x-axis, and here's amygdala response to these fear faces on the y. And what you can readily see is that we get this decline with age. So that means that the younger individuals on average are showing a more robust, a stronger response than older individuals. So the average 5-year-old, for example, has a stronger amygdala response than the average 20-year-old, which might be counterintuitive at first because we often think about brain function as increasing as we get older. But this actually makes sense if we consider that the amygdala's primary job is to learn about the relative safety and danger of the environment. And when you're young and kind of new to this planet, you have a lot of learning to do. So you want the amygdala chugging along at full speed at a very young age. Now we can also look at that superhighway that I talked about between the amygdala and the prefrontal cortex. And to do that, we use a methodology called functional connectivity. Now, functional connectivity is actually a pretty simple measure. What we do is we can extract the activity of one region, like the amygdala, and plot it over time. And then we can extract the activity of, say, the prefrontal cortex and plot it over time. Now, the degree to which these two lines look similar to each other, or the degree to which they're correlated, gives us an index of functional connectivity. It's as if there's a conversation that might be happening between those two regions. Now, it turns out for the amygdala and prefrontal cortex in healthy adults, what we often find is that they're correlated, but they're anti-correlated with each other. So in other words, when the prefrontal cortex starts increasing its activity, the amygdala shows a corresponding decrease in activity. And that's what you often see in regulatory relationships between two brain regions. When one region increases, the other region decreases. In fact, the more negatively correlated the amygdala and the prefrontal cortex are, generally the more emotionally regulated the adult is. So here are data from adults and teens. And what I'm showing you here is that the more negatively correlated the amygdala and prefrontal cortex are, the quieter the amygdala gets over the course of the scanning session. So each dot here is a person. So this individual here has a very negative correlation between amygdala and prefrontal cortex, and their amygdala quiets down a lot over the session. Those individuals also have very low anxiety. Those individuals also have very low sweaty palms, which is how we measure autonomic arousal, nervous system arousal. So the take home from this slide is that typically adults who have very good ability to regulate their emotions tend to have this negative connectivity pattern between amygdala and prefrontal cortex. So now back to our data with 4 to 22-year-olds, we wanted to assess the strength of this connectivity. So what I'm showing you here now is not reactivity of any region. I'm now showing you connectivity between the prefrontal cortex and the amygdala, which you can't see. If you look down here, I'm plotting the data from our young adults, 18 to 22 years old. So they're showing that nice negative connectivity that I just described. Okay, so for them, when prefrontal cortex is increasing, amygdala is decreasing. That's characteristic of this kind of emotionally regulated profile. Okay, so now when we look at the younger and the older teens, they're also showing this negative connectivity, but as you can appreciate, it's not as negative as the adults. So that's consistent with what we know about the slow development of emotion regulation abilities. But what we became very interested in is what we observed in children. So in 4 to 9-year-olds, we don't see that negative correlation, but instead we actually see a positive correlation. So what that means at the very least is that the way that the amygdala and prefrontal cortex are talking to each other during childhood is different than the way that it is during adolescence and adults. And it also suggests that children are not showing this regulated relationship between amygdala and prefrontal cortex. Moreover, these connectivity patterns help explain age-related changes in emotional behaviors. So as individuals start showing this more negative correlated pattern, they also start showing emotional behaviors that they're better able to regulate over time. So to show you in cartoon format what I have been explaining, in the adult state, when we see increases in medial prefrontal cortex, we tend to see decreases in amygdala. So they're working in opposition with each other. But in children, that's not the case. When one region is increasing its activity, the other region is also increasing its activity. This is not a regulated pattern, and these are healthy, typical children. So putting those two findings together, what we're seeing, if this is age on the x-axis here, is that during childhood, we get a very strong amygdala response. But that's happening in the absence of this adult-like connectivity with prefrontal cortex, which we don't see emerging until the beginning of adolescence. So that's made childhood very interesting to us. It means that the nature of the system between amygdala and prefrontal cortex is operating in a fundamentally different way during childhood than it is at points thereafter. It also gives us a moment to focus on for thinking about potential sensitive periods for this system when the environment is going to have a particularly large impact on shaping the way that our emotion regulation neurobiology operates for the rest of our lives. So to formally ask this question about the nature of connectivity at one point in development influencing the way the system's going to work later on. Am I going in and out? No? Okay. To formally ask that question about this prediction over time, what we did was we collected that connectivity measure that I just described in response to fear faces. So this stimulus-elicited connectivity. In all of our subjects, we also collected something called resting state connectivity. So resting state connectivity is the same connectivity measure that I just described to you, but we don't ask participants to do anything. They just lie in the scanner. And what we get is a measure of their kind of functional brain skeleton. So this skeleton in adults is associated with people's ability to regulate their emotions. So we became very interested in how this system constructs itself over time. Okay, so we collected both the stimulus elicited and this resting state measure at time 1. And then we invited everybody back 2 years later and we collected those two measures again. And what we found was the best predictor of this future resting state connectivity, this future functional skeleton of the amygdala prefrontal cortex system, was the stimulus elicited connectivity 2 years ago. So in other words, the way that the amygdala and prefrontal cortex co-activate in response to some stimulus in the environment is a really good predictor of how the system is going to operate even when it's not responding to stimuli, when it's just at rest. Moreover, we looked at the strength of this prediction across age. The younger people were, the better that prediction arrow was. And as you get older, the prediction strength weakens. So what that suggests to us is that the nature of the way amygdala and prefrontal cortex co-activate in response to stimuli is in a way molding the way that the brain is ultimately going to settle into its resting state. So this is consistent with the notion that in childhood we have a more plastic, flexible system that's highly responsive to the environment. And over time that transitions to a more stable system, but that stable system is going to be less amenable to environmental perturbations. So these findings really motivated us to finally ask, is this middle childhood period, the school age period, a sensitive period for the development of these connections between medial prefrontal cortex and amygdala. And we were motivated by many animal studies showing that things that are learned early in life can be used later on in adulthood, often to help reduce anxiety. So I'll explain. We were inspired by colleagues' finding. This is Takao Hensch's work, who's at Harvard. And he found if you place a mouse in the center of this open field, the average adult mouse, and one corner had a nest that was silent, and one corner had a nest that played music. The average adult mouse would prefer to go to the silent nest, right? So in general, mice like to run to the corners of these fields. They're like middle schoolers at their first dance. They just run to the walls. So if they're given the chance to run to a wall, they would choose the silent nest. Unless these mice were pre-exposed to music during their childhood. So if they were pre-exposed to music during their childhood, then they would start showing a preference for the music nest. And the preference was specific. So if they were pre-exposed to Beethoven Symphony No. 1 during their childhood, then they preferred Beethoven Symphony No. 1 and not jazz, and vice versa. And the timing of this was critical. It had to happen during their childhood equivalent. Earlier and later exposures did not have this effect. So not only did they have a preference, but when you played these adult mice their childhood music, it reduced their anxiety. They started to wander out into the center of the field where they're more vulnerable. So when they wander out into the center of the field, that's an indication that they're feeling less anxious. And when they looked at brain activations, they found that the music that comes from childhood would activate cells in the medial prefrontal cortex of adult animals, and it would also reduce amygdala activity. Okay, so we were really excited about this finding because it's actually very hard to identify sensitive periods in humans because we take so long to grow up, right? No grant that we write to the federal government is going to last an entire lifetime. So we need a way to get at a stimulus from someone's childhood when they're adults. And that's very hard because if I were to ask any of you to name a stimulus that you experienced only in childhood and then not again afterwards, it would be really hard for us to do that. And it would be really hard for us as experimenters to know for certain that that was true. So we thought, well, music might give us an avenue for asking this question. Maybe we won't use Beethoven. Maybe we can use pop music. Maybe we can use data from the Billboard charts to know exactly how old somebody must have been when they were bathing their brains in some of this music. So we want a stimulus. If we're bringing in, let's say, a 22-year-old and we tested them in the year 2012, we want a stimulus from when they were children. So we can do the math, and that means that we need a stimulus from 1997. So we searched the archives, and we found a stimulus that would work. So we used the Backstreet Boys for these young adults, okay? So we didn't put people into an open field like the mouse study. We stressed them out a different way. So these were UCLA undergrads, and we gave them hard SAT math. And we watched them over their shoulders, and we told them that they were doing okay, but not as well as the average UCLA student. So they should try and improve their performance a little bit. And it turns out that's very stressful for students. In between the stressor, we gave them breaks. And during the breaks, we told them you could listen to one of two radio stations. One radio station would play the childhood music, so this is Quit Playing Games with My Heart by the Backstreet Boys, and the other radio station would play music from their late adolescent period, so this would be Justin Bieber and Ludacris. And so the question was, which radio station did they tune into more frequently when they were stressed? And so what we found was people, as the number of stress trials increased, people would increasingly choose the childhood music. Our control group were people who didn't grow up in the US and reported not listening to American music. So they weren't choosing the Backstreet Boys, they were choosing Justin Bieber and Ludacris because they were familiar with that. I should point out that this preference here that they're showing is not about liking the music. So nobody's walking in saying that they like the Backstreet Boys. But under stress, under duress, they would show this gravitational pull for the nostalgic childhood music. Okay, so it was only the people who listened to their childhood music that also showed significant decreases in their physiologic arousal during the stressor. And we scanned these individuals. The childhood stimulus was also effective in increasing medial prefrontal cortex activity and decreasing amygdala reactivity. And the more that this prefrontal cortex was activated by the childhood stimulus, the lower the anxiety was for those individuals. So this was a fun study for us to do, but it was also an important one because it gave us a little more confidence that there is merit in thinking about this childhood period as a sensitive period. But we don't really care about what the Backstreet Boys do to your brain. What we really care about are stimuli we're exposed to with regularity that coincide with this putative sensitive period, and that is our caregivers. And so we often think about these brain systems as developing in a test tube or in the scanner, but they're developing in this very important environment. And for humans, when we talk about the caregiver, it can be the biological mother, it can be the father, it can be an adoptive parent, a grandparent, and the list goes on because this is a learned relationship. We learn to form these attachments, as many, many species do. But what is important to point out about humans is exactly how long we spend in this relationship. Right? In most species, it's on the order of a few weeks, maybe a few months that young spend with their caregivers. But in humans, it's decades, right? And that is a huge metabolic investment for a parent to raise another human being. So there's got to be a big payoff. And one of the arguments is that the payoff is that gives our brain the luxury of immaturity. It gives our brain the luxury of this increased plasticity during which we can learn all of the massive things that we have to learn. So in this way, parents are very much forming a scaffolding, like scaffolding on a building, for our central nervous system to give us that luxury of learning. So again, we turn to rodent studies to inspire some of our questions in humans because the rodent work has provided some really beautiful data illustrating this intimate relationship between caregiving and brain development. So this is a summary of the work of my colleague Regina Sullivan, who's down at NYU. And briefly, what she has shown in rat pups is that the functioning of the amygdala right after birth in rats, it's pretty quiet. It doesn't activate too much. But the functioning of the amygdala during adolescence and adulthood functions the way we would expect. It responds to threat, it responds to emotional learning. But there's this period in between where the functioning of the amygdala is critically dependent on the presence of the parent. When the parent is in the nest, I'm going to say she because in rats it's always the mother. When the parent is in the nest, she keeps the amygdala quiet. So in other words, when she's in the nest, the amygdala of this animal functions like a newborn's. And when she's out of the nest, the amygdala increases in activity. So the amygdala of this animal functions like an adult. So the mother's presence is turning on and turning off the amygdala. Now, how this plays out in behavior, I think, is fascinating. So the workhorse protocol of studying amygdala learning is Pavlovian fear conditioning. So if you and I were presented with a peppermint odor and a foot shock, we would all learn that we don't like that peppermint odor. And that's what the rat pups learn when they're not with their mother. So if you place that peppermint odor on one arm of this Y maze, this rat pup will avoid it and go the other direction. And that's what's being shown here. They're avoiding that peppermint odor. But if the learning takes place with the mother present, and the mother is going to keep the amygdala out of the equation, then what happens is this rat pup seemingly paradoxically starts to approach that peppermint odor. So they don't like the foot shock, but if the peppermint odor and foot shock were learned in the mother's presence, they will form a preference. If that sounds strange to you, then you got it right. That's the finding. So you can ask, why would Mother Nature ever design a system where the young animal is approaching this peppermint odor? And the explanation is that this preference learning is critical for the attachment learning that has to happen at this stage in life. So regardless of what this peppermint odor was paired with, it could have been paired with something rewarding, pleasant, or it could have been paired with something aversive, in this case the foot shock. If it's learned in the presence of the mother, then this animal is going to prefer it no matter what. So in other words, for you and me, under threat conditions, our strategies for survival are to fight or flee, but those are useless strategies if you're a young animal. Instead, the survival strategy is attachment learning or preference learning. So we were really interested in this idea that the parent is buffering these stress responses. And I should point out, this needs to be a calm parent, right? So the parent needs to be calm in order for this effect to happen. So we and others wanted to know, given that human development is so stretched out compared to a rat, might we see evidence of this buffering in childhood? So these are data from Megan Gunner's group in Minnesota, and they asked children to come in and prepare for a public speech, which is a wonderful way to elevate your stress hormones, in particular cortisol. And children, when they're preparing for their speech and they're sitting next to a stranger, they show this nice increase in cortisol over time, just like you and I would. But if they sit next to their parents while they're preparing, then instead that cortisol response is buffered. They don't show that same elevation. Now, adolescents don't show that buffering. Adolescents would show this nice, beautiful cortisol response regardless of who they're sitting next to. So there's something about children here. So we collaborated with Regina Sullivan, and we just did that conditioning experiment in children. So here we presented children with a blue square, and it coincided with not a shock, but instead a terrible noise, which I'm going to play for you, but it's like nails down a chalkboard. And the triangle was paired with nothing. So children learn these associations sitting next to a friendly experimenter or sitting next to their parent. And their parent wasn't doing anything, they were just sitting calmly. Following these pairings, the parent went away, and we built a human Y maze. And we had children enter into a tent, and we placed the blue square here, the purple triangle there, and we asked children to crawl in and then pick a door to retrieve a prize. And we showed them that it was the same bucket of prizes behind both doors. We just wanted them to pick a door. Okay, so now I'm going to show you what children saw, and just a warning that the blue square is going to come up with that noise, but I don't think it's too loud. Okay, so they would see this, and then it would go away, and this, and then it would go away, and then this, and then it would have that and go away, and then this, right? So it's terrible. It's not painful, but it's terrible. Okay. So now, just like we saw in the rat study, we wanted to assess whether children were preferring or avoiding that blue square. So this is in 3, 4, and 5-year-olds. When they conditioned sitting next to the experimenter, they more or less did what you and I would do. They avoided that blue square in the tent, in the Y maze. But when these same children were sitting next to their parents and during conditioning, then now the parents seem to be blocking that aversion learning, and we get a slight but significant preference for that blue square as well. And I want to point out that this is a within subjects design, so these are the same children whose behavior, their learning is switching as a function of who they were sitting next to. And we know in the rat that this effect terminates developmentally as the stress hormone cort starts to show its developmental increases. So we collected saliva cortisol, and it's, again, a modest but significant association such that children who were producing more mature levels of cort were less likely to show a preference for that blue square. In other words, they were less affected by their parents' presence. They were more acting like you and I would, suggesting that there are biological constraints to this effect. Okay, we also scanned children and we presented them with pictures of their parents to see if presentation of a parental cue would have a similar effect at the level of the amygdala. So we presented children with pictures of their own parents and contrasted that to pictures of somebody else's parents. And what we observe is that in children, pictures of their own parents were now dampening the amygdala response. So we're interpreting this like the rat data, as the parent seems to be buffering some of these systems. We don't see that same modulation or that same change in adolescence. And it's not to say that parents aren't important during adolescence, they certainly are. But it may be that at the level of the amygdala, the parent has started to lose some of his or her effect on the system. And now we tested that superhighway connectivity between amygdala and prefrontal cortex. And so this is the connectivity data between the prefrontal cortex and the amygdala, which you can't see. And here we see that presentation of the parent is strengthening that negative connectivity with prefrontal cortex. So in other words, at the beginning of the talk, I showed you that children were showing this non-regulatory connectivity pattern between amygdala and prefrontal cortex. But when we present the parent cue, then we start to at least momentarily see that top-down type of control from prefrontal cortex. So the parent cues are temporarily giving us these more mature regulatory connectivity patterns. And the strength of this effect was even stronger for children who reported secure attachment relationships. Okay, so I'm almost at the end here, if I'm okay on timing. So that was all the effects that we've seen when parents were able to be calm or appear calm. But we also know from the rodent data that parents' own anxiety or own fear behaviors are also really effective in amplifying amygdala reactivity and stress reactivity. So to ask whether the same would be true in humans, we now had children watch their parents undergo fear conditioning. So this is me, for example, getting fear conditioned. This circle would always be paired with that terrible noise for me. We would record me and then show it to my daughter. So she wasn't conditioned herself, but she saw me experiencing the conditioning versus somebody else's parent experiencing the conditioning. Then in the scanner, we showed her either the stimuli I conditioned to or the stimuli that someone else's parent conditioned to. And what we find is that children learn better. So down means they are learning better. They dislike that stimulus more. They learn better to dislike the stimulus that their parent conditioned to, even more than somebody else's parent. They still don't like it, but the effect is stronger for parents. And the stimuli that I learned was more effective in increasing my daughter's amygdala than the stimulus that somebody else's parent was conditioned to. So taking these data all together, what we think is happening is that during childhood, we have parents having a lot of control over the emotional neurobiology of children's brains. And so we are thinking that this is a sensitive period for the toning of this system, where parents are able to both dampen as well as increase amygdala reactivity. And we think that that is important, that dampening as well as that increase is helping to teach the system or entrain the system about the boundaries of emotional experiences. And that learning involves a cascade of neural and hormonal events that change behavior during childhood. But the learning that happens during this putative sensitive period ends up influencing the nature of the system at points thereafter, even once this sensitive period closes. So we're thinking of parents during childhood as providing this privileged information source about the emotional world. So to conclude, we see that brain areas that support emotion regulation develop very slowly in humans, but we think that slowness has great value, that there's great value in slow cooking of the human brain. And this transition to the adult-like brain within this circuitry seems to emerge at the beginning of adolescence, but continues to show a lot of developmental change at points thereafter. So this gives us a lot of time for the environment to scaffold. Now, I know I focused a lot on parents, but given that there are a lot of teachers in the room, we would also predict that teachers are doing a lot of this scaffolding, a lot of this modeling that is influencing brain development. And parents are really serving this primary social regulator role at a time when the brain isn't so good at doing it on its own. Well, we're lucky as a species because we have parents that can help us do that as we're developing. And parents provide this privileged information source. So I think that this has a lot of implications for the way that we as a society think about the future as well. So if we care about the future, we need to care about children and adolescents. And given that children and adolescents aren't growing up in isolation, but they're growing up in this very special environment of the early caregiving environment, then we also need to be thinking about how do we support families in order to do this very difficult and metabolically costly endeavor of raising another human being. So I think that there are a lot of implications for what we want to think about doing as a society. So with that, I'll just thank the lab, of course, and the funding sources, but also especially, the families and individuals that participated, and I want to thank you for your attention. And I'm happy to take questions. Okay.
Thank you very much for your lecture. Most interesting. I've been reading a book, still reading a book called The Biology of Behavior, Good and Bad. I forget the author's name. He indicates that during this period that you're talking about, from early childhood towards the early 20s, there is something called dendritic pruning of the neurons. What's your take on that?
Yeah, well, that's a great question. So in general, what we see in brain development is what I think is a fascinating process where we actually initially overproduce the number of brain cells that we're going to need, and we overproduce the number of connections between those brain cells. So in other words, a young infant has more brain cells and more connections than we do as adults. And that sounds really metabolically expensive. So why would you ever design a brain that way? And that is all related to this idea of brain plasticity. So the reason is, or the theorized reason is that we don't know what household we're going to land in. We don't know what language our family is going to speak. We don't know what food and diet we're going to be eating, so on and so on. So this incredible number of synapses that we have and cells that we have allow us to be able to fit into any of those environments. And then as we learn from the environment, as we hear what language our parents are speaking, then we prune these synapses, or dendritic pruning as you're describing, to get rid of the stuff that we don't need and really home in on the synapses that we're going to want. So it's really a beautiful model of how the environment, the early environment, is really literally sculpting our brain development.
My question perhaps may be related to that. Children from the ages of birth to the third or fourth year experience a lot of very specific events. And if you talk to them at that time period as to what happened last week, their prior birthday, they can tell you. Some years later, there's this mass extension of all specific memory of any particular event. Does that mass extension serve a purpose, and is it related at all to the first part of your lecture?
That's a great question. So what you're describing is commonly referred to as infantile amnesia or childhood amnesia. And it's a hot area of research right now, and there's a trainee in my lab that is specifically interested in this. So it's unclear exactly why this happens, because what you described is exactly right. It's not that children aren't remembering things. It's just that, in fact, sometimes children have exquisite memories when they're at that age. They can remember details that I'm like, how did you even remember or notice that? But then when you ask them again when they're 13, they have no memory of it, right? And in general, people say that on average, most of us can remember, our earliest memories are around like 3 to 5 years old. And so there are a number of hypotheses about why we experience infantile amnesia, and we're not the only species that experiences infantile amnesia. And some of the ideas have to do with the development of the connections between, for example, hippocampus, which is playing a central role in binding memories for us, and its communication to these higher order prefrontal regions, that there is a massive change over childhood, and that change is kind of repackaging the information in a way that's very difficult for us to access declaratively as adults. But it's a fascinating topic.
Thank you for the fascinating lecture. I wanted to ask a question about the relationship between amygdala and prefrontal cortex, which, as you explained, sort of in the more mature state of adults, has the negative relationship where, increased activity of amygdala could be brought down by well-working prefrontal cortex, and the aspect of presence of calm parents with children, helping regulate this in children where when they cannot do it on their own, which led me to conclusion that sort of presence of calm parents in childhood helps develop this relationship towards the more regulated state in the adulthood. And so I guess my question would be, what are your thoughts about impact of highly agitated parents or presence of parents which are not calming at all, or maybe even parents who create the terror in children's lives?
Right. So it's a really important question and related to my thoughts about how do we help children. And I think that the way that we help children is to help support families, right? So for children who might be struggling with anxiety, for example, one of the treatments that people use particularly for young kids, is to involve the parent, right? So to help the parent identify what are some of the reactions that they might have, even with the best of intentions, that could be exacerbating some of those behaviors. So I think that it's really difficult to think about the child in isolation. So there's Winnicott is an analyst from many years ago, and he famously said, there's no such thing as an infant, which is to say that it's always the infant and parent. And I think the same, we can extend that further into childhood, that there's no such thing as a child, it's the child and the family that they're living in, and the society that the family's living in, and so on.
Okay.
Okay.
Yes, go ahead. Hi. I really like the lecture, by the way.
Thanks.
So since you said like we can't pick, like when we're born, we can't pick the environment that we're born into, how will the connectivity measure between the prefrontal cortex and the amygdala, I don't know if I said that right, change when a child suffers through a traumatic state or time, like a car crash or their family dies or some like bad tragic event?
Yeah, that's a good question. So I didn't present it tonight, but a lot of the work in our laboratory is very interested in early traumas, particularly early caregiving traumas related to your question. And what we're seeing and what's been seen, again, across many species is the finding is kind of counterintuitive. So the finding, generally speaking, is that we often think about stress as impairing function. That's often true when you look in adulthood. But when you're looking during development, what people often find, including us, is that there's actually an acceleration of development of these circuits. So you start to see more adult-like function. And so it's as if it's akin to like needing to grow up quickly in order to manage bigger emotions, more adult-like things. And so what we don't know is that a resilient thing? Is that a good thing in the long run? Or is that something that we want to prevent from growing up too quickly? But when we first came across that finding, it was counterintuitive to us because there's so much in the literature that is written as if stress is going to impair, but instead during development we see that there seems to be some kind of facilitation, at least within this circuitry. It's a good question.
Thank you so much for your lecture. So my question is, at the beginning of the presentation, you mentioned mental illnesses, and I was just wondering, like, first of all, how does early exposure to, like, parents with mental illnesses inform future, like, exposure and development of mental illnesses in the children in their adulthood, and whether or not, like, these learned behaviors from this environment that you didn't choose to be in, like, whether that can be changed even though like in adulthood the brain is less plastic, and also like what are the ethical implications in terms of like would you be able to tell, like you can't just tell someone with mental illness that they can't like raise a child, and that's my question.
Yeah, so again, we definitely want to help parents be healthy and happy as well, because that will influence children's understanding of the emotional world. So that I think I would put at the center of things that we want to think about for interventions for young kids. The second question you asked is one that many people, I'm involved in a network that we call ourselves, in quotes, reversibility network. So the idea is, if plasticity is so high early in life and now you're an adult, what can we do? How can we think about changing behavior and brain function? And I think there are a lot of ways we can think about doing that, but brain plasticity doesn't, at least in the human, doesn't seem to disappear. It just seems to decline relative to the massive amount of plasticity that we have early in life. But we know that brain plasticity doesn't disappear because we continue to learn as adults. We take classes, we go to lectures, and so there's a lot of work that's focusing on wellness behaviors in adults, like exercise, sleep, good nutrition, and so on, and ways to kind of activate that circuitry. And certainly in areas that focus on cognitive decline and aging, there's a lot of emphasis on engaging in novel activities and really kind of working that plasticity again.
And what about the ethical implications? Because I think that's something that's very important in society right now, because a lot of people, like a lot of adults are exposed to mental illness, and that does inform the way their children grow up. But how, like, there is not really what is a solution that would change, I guess, the environment of parenting without like telling like parents that they can't have children?
Right. So we're not going to do that.
Yeah.
But I think that finding ways to support adults is a primary step in helping children. So that means lots of different things for different populations. So that support could be mental health support, but that support could also be financial to reduce stress that families are experiencing. That support could be reducing injustices in our society and discrimination and so on. So I think that these are very large issues that isn't boiled down to one individual. I really think it's us as a society issue.
Okay, thank you.
Hi.
Hi.
Good evening. Thank you for your lecture so much. And just out of curiosity, whether it being scientifically or biologically speaking, the transitioning from somebody's childhood, based upon like recent discoveries and recent studies, the connection between parental and their child, like a connection relationship, it seems that the child really depends on the parent, whether being in a stressful situation and a stressful, like, I guess, environment they're exposed to, they're always attached to this parent. And like I've always wondered where does this rebellious, I guess, action come from a child, whether it being politically rebellious, whether it be religious rebellious, whether it be like just going against, I guess, parental control and parental like guidance that such a wise person would do. Like if the child had like, I guess, a third eye, quote unquote, open, they would see that that's like what the best. Yeah. That's what the parents want the best. Like where does this come from?
Yeah. So. Are you the parent of a teenager?
Yeah, well, oh no. Wait,
so a lot of researchers are interested in exactly that question, and the way that I ask that question is, if we didn't rebel against our parents, and some people rebel a lot and some people rebel a little bit, if we didn't rebel against our parents, we might not be here as a species, because we need to want to leave the nest, right? So we can start our own families and so on. So there's got to be a big pull out of the nest, because why would anyone want to leave home? You're being fed. People love you unconditionally. You're not paying rent, right? So that's all good stuff. So you've got to build something into our biology that wants us to get out of that. And so that's where a lot of the adolescent brain development that focuses on sensation seeking and reward learning comes into play. So we get a nice big bump in those regions of the brain during adolescence. And so it can come out in rebellious behavior, but those systems also support a lot of the things in adolescence that you don't see at earlier points. So an interest in new political ideals, civic engagement, passions developing, art appreciation, and so on. So those are probably all bundled together in the thing that gets us out of the house, even if it kind of drives parents crazy a little bit.
I'm sorry, can I ask another question? Is that right? Oh, I'm sorry. It's all right.
We can talk afterwards. Okay. Yeah.
My question is, so a lot of the data that you shared with us involved the primary caregiver.
Mm-hmm.
Is there anything comparing primary versus like maybe secondary, or the relationship besides secure attachment style?
Yeah, so we haven't done that yet, and I haven't seen other labs do that yet, but believe that there is something coming down the pipeline that's asking that question. With that, we used a really blunt instrument here, right? Like parent, stranger. So we would want to look at other forms of attachments that could be the other parent. From teachers always ask us, what about the teacher? People want to know about friends. So what role can friends be a good buffer? So we don't have the answers to those things yet, but there are data from UCLA out of Naomi Eisenberger's lab that shows in adults, romantic partners seem to be playing a similar type of buffering role. So there's room to think that it's not just the parent, but the parent's doing a lot.
Good evening. So my question is, well, we as species, we have this genetic memory, and we think, first of all, we think to survive. You know, we have all these things that help us survive, but then this environment and family and society, everything that actually we don't choose. For example, I don't choose the family that I'm in, so I identify myself with that family and the society and environment and everything. But then, is there a way that maybe when, like in adulthood, is there a way that I can disconnect my brain from that memory and from everything, from the environment and everything, so I can think in a creative way to ask or to answer a question that doesn't have to do with what I'm surrounded of? Like maybe even for discovery, like something a way to disconnect from everything and just find my own way, answer a question or whatever it is.
Yeah. I don't know the answer to that. I wish I did for my own sake. But you're asking a question that's sort of out of my wheelhouse. But I suspect that folks that are doing research on things like mindfulness or meditation are kind of asking that question, is how to remove some of the chatter in order to focus on that quiet voice a little bit more. So that would be the literature I would recommend.
Okay, thank you.
Thanks.
Could you be more specific about childhood sensitivity in the time period? Because a lot of people say, give me the child until he's 6 and I'll give you the adult.
Yeah.
Is that true, or is it go further into adolescence?
Yeah, we really are interested in that question as well. So in our data so far, what we generally see is 9 is about the end of childhood in our hands. I suspect that is far from a hard number, that there's a lot of error around 9, and there's going to be differences between boys and girls and differences from one culture to another, differences as a function of early life stress. But in our data, which are taken from metropolitan-based American kids, so I don't know how much that generalizes.
Yeah. And the second question I had was about caregiver versus mother, or as we have more, 60% of couples are both working and they have nannies now. Do you know is there any data on that?
So my take, and then I'll give you some data. My take is that the role of the primary caregiver, that really is a learned role. So yes, it's often the mother, but it doesn't have to be. It could be the father, it could be the grandparent, and so on. And there are, arrangements where you could have multiple primary caregivers within one family, right? So there's all different kinds of families. So it's learned from the child, but it also seems to be learned by the parent. So there are data, Ruth Feldman, who is a researcher in Israel, has this paper I really like where she first scanned the brains of mothers and fathers. And in this study, the mothers were all the primary caregivers. And she showed that, mothers' brains are responding to baby cries in a very special way. She then followed that up with another study where both parents were dads. And one of the dads, who was he identified himself as the primary caregiver, had the same brain response that the moms had in the other study, really suggesting that learning how to have a brain response like a parent is learned just as much as learning how to attach to a particular parent is learned by the child.
So thank you very much for the lecture. It was really interesting. And so I'll just cut right to the chase. So there have been recently studies that claim that the brains of schizophrenics contain more flexible neurons. And so one thing, so is that the same flexibility that you're talking about when it comes to children's brains?
So it's a great question. Your question is actually related to a couple of the earlier questions about pruning, and one of the theories is that there is a difference in pruning of neurons in individuals living with schizophrenia. I think it's a really good question about whether what we're seeing is similar to the plasticity that we see during childhood. The mouse study that I showed you, where the mice had this kind of critical, sensitive period for the music, that researcher has asked that exact same question and has made animal models that are genetically showing similar phenotypes, similar outcomes as humans living with schizophrenia. And what he's found is that you get that same type of plasticity in those animal models. So it suggests there's merit to that hypothesis.
So you just answered my second question right there. Thank you.
Mm-hmm.
The chicken egg question is whether adolescence starts first or the moving away and changing starts first. So I'm interested in your view on that. And the other piece is, did you notice any gender differences in your studies?
So I'll answer the second one first. So we did not see gender differences, but to be fair, these studies weren't adequately designed to ask that question because you need big studies, and we're doing that right now. That being said, I wouldn't necessarily expect gender differences in these fundamental attachment learning processes because we all have to attach, but I might expect them more as the individuals age and pass puberty. So to your adolescent question, there are currently a lot of conversations back and forth in the adolescent world asking exactly that. And one definition that I keep hearing again and again is that adolescence seems to be something that starts with a biological definition and ends with a cultural definition. And so depending on when people start taking on adult roles, which for some people can be 15, for other people it can be 28, that kind of marks the end of adolescence. So we do look at puberty in all of our studies. We also collect testosterone levels and so on. And there are some effects that we see that are related to puberty, but overwhelmingly what we're seeing is they seem to be more related in the things we're looking at to environmental changes. Thank you.
So, thank you, Nim, for this fabulous lecture. And thank you all for coming. Thank you again for the Niarchos Foundation for the sponsorship. So we appreciate your help and partnership. We hope you can continue to come and help us be connected to society. I think a lot of what you hear really has real life implications. I was thinking a lot about my own family and me as a parent or as a kid. So we have more of these events. The next one you'll see is going to be the first one at the Columbia University Forum at Broadway and 125th, and will be given by Tom Maniatis, and he's going to tell us how we can now use genetics of single cells to find the identity of the billions of neurons we have in the brain and how that's important for normal function and disease. Again, thank you, Neem, and thank you all, and have a safe journey home. Thank you.
Yeah.
Oh, yeah, but, can you make